Membrane potentials. I will be talking about two types of membrane potentials in this lecture. One is resting membrane potential, which is the difference in electrical potential between the interior and exterior of a biological cell. The other is action potentials, which is a rapid change in the membrane potential that spreads rapidly along a nerve fiber, facilitating a nerve impulse. Before I get into the details of membrane potential, I want to give a brief summary of a membrane receiving a nerve signal. When the neuron membrane is at resting membrane potential, it is considered polarized. At this state, the neuron can receive a stimulus. The stimulus will cause membrane ion channels to open. When the channels open, sodium will flood into the neuron. Sodium has a positive charge on it. So the influx of sodium will change the electrical potential across that neuron's membrane, depolarizing it. This depolarization can sweep along the neuron's membrane by opening voltage-gated ion channels, and this is called propagation. Electricity in the body. The human body is essentially neutral. However, just around the membrane, there is a charge on just the outside of the membrane and just the inside of the membrane of all cells in our body. The outside of the membrane is predominantly positive when the cell is at rest, and the inside of the membrane is predominantly negative when the cell is at rest. Voltage, current, and resistance in the body. Just some definitions I want to cover before we go into too much detail. Voltage is the difference in electrical potential energy across a cell membrane. So it's referring to the charge that is just inside the membrane and the charge just outside the membrane. If you look at the diagram on the right, you can see that the voltage of a neuron between the outside of the membrane and the inside of the membrane is negative 70 millivolts when that membrane is at rest. Current is the flow of electrons across the cell membrane. That current would be the sodium ion channels entering the neuron when channels get opened. Resistance is the cell membrane itself, depending on how many protein channels it has and how those protein channels are activated. The length and diameter of an axon also can present resistance. The longer an axon is, the longer it will take for a signal to reach its destination, so that would be resistance. Also the diameter, if an axon is very narrow or gets narrow in certain parts, that'll provide resistance for that signal to travel down the axon. Resting membrane potential. At rest, a neuron cell membrane is considered polarized. As you saw in the diagram on the previous slide, it is at negative 70 millivolts when it is at rest. That means it is slightly more negative on the inside of the membrane compared to the outside of the membrane. This means that the resting membrane possesses electrical potential, meaning it is ready to receive a stimulus. Any cell membrane that is not at its normal resting membrane potential cannot respond to a stimulus. If a neuron cell membrane has a resting membrane potential, it will also have a threshold. The threshold for neurons is negative 55 millivolts. So any event that causes the membrane potential to reach threshold will facilitate an action potential. If we look down at the chart, you'll see that it starts with the membrane at negative 70 millivolts, which is resting membrane potential. If a stimulus opens up the sodium channels, allowing an influx of that positive ion, it'll cause that membrane to move closer to zero. And once that membrane reaches negative 55 millivolts, it will facilitate an action potential as you see it peak at number 3. It'll peak at positive 30 millivolts, and as soon as it peaks, it starts to repolarize, as you can see on the back side of that curve. Resting membrane potential is dependent on establishing and maintaining the ion concentration inside and outside of the cell. It is this ion concentration that maintains the negative and positive charges just on either side of the membrane, respectively. Normal ionic concentration is a high concentration of sodium outside the cell with a low concentration of sodium inside the cell. It is opposite for potassium. Potassium will have a high concentration inside the cell and a low concentration outside the cell. Resting membrane potential is established and maintained by the sodium-potassium pump. If you remember, this pump is an active transport pump requiring energy. It needs the energy because it is continuously pumping 3 sodium ions into the cell and 2 potassium ions out of the cell against their concentration gradient. So why does every single cell in our body need to have a sodium-potassium pump in its membrane? It is because all cells also have leaky channels that allow passive movement of sodium and potassium in and out of the cell down their concentration gradients. This passive movement will continue until the ions reach equilibrium. However, as long as the sodium-potassium pump is working, they'll never reach equilibrium because it's always pumping those same ions against their concentration gradient. This constant movement of ions is essential for maintaining that negative 70 millivolt resting membrane potential. The anatomical makeup of cell membranes is that there are more potassium leaky channels than there are sodium leaky channels. So this concentration of potassium, leaky channels, makes potassium the #1 ion involved with establishing resting membrane potential, and sodium is the second ion involved in establishing and maintaining resting membrane potential. What prevents all the potassium from leaking out? It is called the Gibbs-Donnan equilibrium, where a negative charge of a non-diffusible ion hinders the diffusion of a cation. What this means is that inside the cell, there are amino acids, and these amino acids are large and they can't get out, and they have a very high affinity for the potassium. So the potassium will bind with all these free amino acids. Therefore, only unbound free potassium ions will actually diffuse out of the cell membrane through these leaky channels. This will cause an uneven distribution of the ions across the cell membrane, which we already talked about before, that there's a higher concentration of potassium inside the cell than there is outside, and that uneven distribution of these anions are essential for maintaining resting membrane potential. And this slide here is just a recap of how neurons maintain resting membrane potential. They do it with the ionic diffusion that I just talked about, where the ions will diffuse through leaky channels down their concentration gradient. They do it by the sodium-potassium pump that moves the ions against their electrochemical gradient. And then the Gibbs-Donnan equilibrium with the amino acids holding on to or binding with potassium, only allowing free potassium ions to go through the leaky channels. Changing membrane potential allows a neuron to receive, integrate, and send information. And this all requires the opening of ion channels on the membrane that is supposed to receive the signal. What events opens ion-gated channels? Well, there's a bunch of them. A neuron will respond to electrical stimuli, chemical stimuli, and receptor stimuli. The electrical stimuli will open voltage-gated channels. Chemical stimuli will get opened by either neurotransmitters or hormones binding to ligand-gated channels. And receptor stimuli are mechanical, thermal, and chemical receptors that are in our skin and in our organs, like your Merkel cells, and we have free nerve endings in our skin. We also have mechanical receptors, say in our muscles. When you have a muscle spasm, that's eliciting a mechanical receptor. The chemical receptors monitor chemistry in our blood, so don't get that confused with a chemical stimuli of a neurotransmitter or a hormone. Chemical receptor stimuli is looking for things changing in the chemistry of our body, where if it gets too acidic or too basic and stuff like that, or uh, chemicals that are released when you have inflammation. That's a big factor in stimulating a chemical receptor. Sodium ion channel densities. Neurons have a lot of sodium channels. Nerve conduction is dependent on the opening of sodium channels. As we talked before, it is the sodium entering in a cell membrane that is the biggest factor in changing the membrane potential. There are some spots on the neurons that have higher numbers of these sodium ion channels than other areas. The highest number of sodium ion channels is located at the nodes of Ranvier and the initial segment of an axon. That's why those areas are considered more electrosensitive. Hyperkalemia effect on resting membrane potential. In the situation of hyperkalemia, that means there's more potassium in the extracellular fluid than there is on the intracellular fluid. And since we know that ions like to move from an area of high concentration to low concentration, you can see by the diagram that potassium is not going to want to move through those passive ion leaky channels because there's already too much out there. So you'll either have no efflux or very little efflux. This is going to affect the resting membrane potential in that it'll make it more positive on the inside. This changes the membrane potential to where it's going to move closer to threshold. When the membrane is closer to threshold, it's going to be more excitable. It won't take as strong of a stimulus for it to reach an action potential. So it's going to be a bit hyperactive. Hypokalemia effect on resting membrane potential. This is the opposite situation. This means that there's very little potassium in the extracellular fluid, and so you'll have a lot of efflux. Stuff will move from high concentration to low concentration, so you're going to have a rapid efflux of potassium exiting the cell, which will make it more negative on the inside. This can cause a hyperpolarized state of the neuron, meaning that it's moved further away from zero, and then it will require a much stronger stimulus to generate an action potential. How does calcium affect sodium channels? The extracellular calcium levels significantly affect voltage required to activate or open voltage-gated sodium channels. Calcium will bind to the exterior surface of a voltage-gated sodium channel. The positive charges on the calcium will alter the electrical state of these channels, requiring a higher voltage to open them. Hypocalcemia, that means low blood calcium levels, will increase the excitability of nerves and muscle tissues because it decreases the amount of membrane depolarization necessary to open the voltage-gated sodium channels because you don't have very many sodium ion channels surrounding the exterior surface of the voltage-gated sodium channel. The membrane does not have to actually reach threshold to propagate an action potential. Hypercalcemia will have the opposite effect. It decreases the excitability of sodium channels because you'll have a lot of sodium ion channels on the exterior surface of the voltage-gated sodium channel. So it will require a higher voltage to open these voltage-gated sodium channels. It will require a stronger stimulus to propagate the action potential. What are action potentials? They are electrical signals that transmit down the length of a neuron's axon. How are action potentials generated? Well, first you have to change the voltage across the cell membrane. Voltage will change when a channel is open and sodium channel moves across the cell membrane to the interior, causing it to be more positive on the inside than it is on the outside. This change in voltage will change the polarity of the neuron, and once the polarity changes to where it is depolarized, it will facilitate an action potential. These action potentials are transmitted or conducted along the axon from the cell body towards the axon terminal. They are self-propagating, which we'll talk about in a little bit, how the action potential itself opens and closes channels along the axon so it can continue to move. Phases of an action potential. It starts with the cell membrane at resting membrane potential and ends with it at resting membrane potential. So the resting membrane potential of a neuron cell body is called polarized. That's where it's negative on the inside and positive on the outside. That means it's ready to receive a stimulus. So once it receives a stimulus, phase 2, it'll initiate local depolarization. This depolarization wave will spread, and if it's strong enough, it will cause depolarization of the entire neuron, which is phase 3. This depolarization will generate an action potential. Phase 4 is where the action potential itself now is self-propagating. That means that action potential will change the polarity of the neuron membrane as it moves down from the cell body towards the axon terminal. Once it has passed a segment of the neuron, that segment will then repolarize, and that segment will return to resting membrane potential. Action potential, amplitude, and velocity. Once generated, an action potential maintains its constant amplitude and velocity as it propagates down the axon all the way to the telodendra. Changes in membrane potential relative to the permeability to sodium and potassium ions during an action potential. For a neuron to generate and propagate an action potential, the cell membrane must become less negative on the inside. It has to move closer to zero to depolarize. Generating and propagating action potential requires the opening of sodium channels and the closing of potassium channels. If you look at the graph on the right, you can see the bar across the top will show you which channels are open and which channels are closed during each phase of an action potential. Here are just some terms: depolarization versus hyperpolarization. Depolarization means the inside of the membrane becomes less negative. It moves closer to zero. A simple example is it'll go from resting membrane potential of negative 70 millivolts to negative 65 millivolts. Hyperpolarization is the opposite. The inside of the membrane becomes more negative. It moves further away from zero. So the example would be at resting membrane potential of negative 70 millivolts. Hyperpolarization could move it to negative 75 millivolts. Polarity changes during an action potential. First, the resting membrane potential cell membrane is polarized at negative 70 millivolts. Then you'll have a stimulus that'll open protein channels, allowing an influx of sodium ions, making it less negative inside, and the membrane becomes depolarized, or depolarization begins. That'll allow the membrane to reach threshold at negative 55 millivolts. Once it reaches threshold, an action potential will occur, and it'll peak at positive 30 millivolts. Once it peaks, it'll then close the sodium channels and open potassium channels. The potassium will flood out through leaky channels, allowing it to become more negative on the inside, and repolarization begins. The action potential repolarization will overshoot the mark. It always does. So it will become more negative than resting membrane potential. But soon, the sodium-potassium pump will quickly restore it back to resting membrane potential at negative 70 millivolts, and then the membrane is polarized again. Action potentials have two refractory periods: an absolute refractory period and a relative refractory period. The absolute refractory period is from when the threshold is reached at negative 55 millivolts until repolarization is about 1/3 complete. So it's really almost the entire action potential. The axon cannot respond to an additional stimulus during this absolute refractory period. The relative refractory period starts from the end of the absolute period until the nerve returns to resting membrane potential. Action potentials can be generated in this period if the stimulus is really strong enough or stronger than normal, because this period the membrane is actually hyperpolarized during the relative refractory period. Graded potentials. Neurons are constantly bombarded with signals. Some are stimulatory and some are inhibitory. The accumulation of stimulatory signals must be greater than the accumulation of inhibitory signals for a neuron to generate an action potential. And it does this by grading the potentials. Types of graded potential. There's local responses that cause a local depolarization, which means it was subthreshold. The stimulus was not strong enough to generate an action potential. This is also known as excitatory postsynaptic potentials when it occurs at a receiving neuron. The other type is an electrical potential, which causes local hyperpolarization, making the membrane more negative, thus causing it harder to respond to a stimulus and harder to generate an action potential. It is also called an inhibitory postsynaptic potential if this occurs at a receiving neuron. Subthreshold responses are local responses. The local membrane responds to the stimulus, moving the inside of the membrane a little closer to zero, but it doesn't reach threshold. And if it doesn't reach threshold, you will not have an action potential. You will not have a current go down to the axon terminal. It will not propagate. Nothing will happen. Conduction with decrement is a term to describe what occurs with subthreshold responses. The current flow that decreases in magnitude with distance is conduction with decrement. You'll have no propagation. Threshold is not reached. This could be because the stimulus was not strong enough to open enough sodium channels, or the membrane was already hyperpolarized at the time of the stimulus. Repeated local responses. We already talked about how a local response can make the resting membrane potential less negative, how it can move it closer to zero. Local responses do not have a refractory period. That means they can be summated. If the subsequent stimulus occurs before it reaches resting membrane potential. So repeated, rapid, local responses can lead to an action potential. Electronic potentials cause local hyperpolarization, which makes the membrane more negative on the inside. This can be caused by inhibiting neurotransmitters such as dopamine, serotonin, gamma-aminobutyric acid, also known as GABA. Action potentials are known as an all or none event. Once a neuron cell membrane reaches threshold, it will generate an action potential. Once that action potential is generated, it cannot be stopped. An action potential typically propagates away from the cell body towards the axon terminal, and it will not die out with distance. It will maintain its strength and speed of signal all the way down, and its amplitude and velocity will be consistent all the way down the axon until it reaches the telodendron. How do action potentials propagate? Action potentials travel down an axon fiber section by section, maintaining a constant amplitude and velocity. Each section depolarizes and then repolarizes. An action potential will open voltage-gated sodium channels at each section, making the membrane more positive on the inside, causing depolarization. After the action potential peaks, it will close the voltage-gated sodium channels and then open voltage-gated potassium channels, making the membrane more negative inside, causing that section to repolarize. Variations in impulses. The intensity of a message or nerve impulse is determined by how many action potentials are generated within a given time span. Once a section of the axon has repolarized, it can respond to another signal. A neuron can respond to a subsequent signal before the preceding signal reaches the axon terminal. Neurons can rapid fire action potentials. A mammalian neuron can receive and or send approximately 500 impulses per second. If you look at the diagram on the right, you'll see the first one, it's showing the action potential in blue, where sodium is influx and changes the polarity. The second one, it shows that the the sodium or the depolarization has moved to another section, and the previous one is now green because it has open potassium channels. Now on the third one, it shows that the very first section that we looked at that was depolarized is now back to resting membrane potential. So that little section right there can receive another stimulus. Myelinated versus unmyelinated axons. Unmyelinated axons are slow. Each section of the axon must depolarize. Action potentials get slower as they propagate. Myelinated axons are fast. Only the unmyelinated sections must depolarize. Therefore, action potentials can zip through the myelinated portion of an axon, and this is called saltatory conduction. In myelinated axons, the action potential maintains its speed.